is by donating their data. And we did just open up the Insight Biobank. This gives you another opportunity to give huge, I can't even put a dollar amount on the huge donation that this would be by donating tissue, blood, other fluids that we're collecting, as well as medical records. Because this is going to give science a look into what makes your uveal melanoma unique to you. And they can look back and say, hey, this cell line, this person's had this, this, and this. Let's try that. Or hey, this person has a medical record of maybe 3 or 4 autoimmune disease. Was that a trigger? And it gives people more information where scientists can start thinking differently because they have more tools to work with. So we're really excited to launch our biobank with Pattern.org. Pattern.org is an initiative of Rare Cancer Research Foundation, and we have Marshall here today who's going to talk to us about the data and how it works and what they're doing over at Pattern.
Thank you. Hi, everybody. You just said it all, right? I don't know if you need me to. That was perfect. You need a marketing job. But I'm happy to talk about both the Cure Insight Biobank and why donation of tissue and data is so important. And so I have a few slides, and then we'll have plenty of time for Q&A, and I'm delighted to talk about any of this. By way of background, I did my PhD in genetics, actually, down the road here at Duke. And then I've spent a lot of my career bouncing back and forth between life sciences and software. So I've been a bioinformatician, actually trying to use this data to find interesting and important things about cancer, as well as building software tools to help other bioinformaticians do exactly that. So I've been at the Rare Cancer Research Foundation for almost 3 years. We were actually founded by a uveal melanoma patient. I think many of you might have even seen his story. It's up on our website. Mark Labs is his name. And the goal of the foundation is to provide a brighter future for folks with any rare cancer, right? So we're pan-rare cancer, but obviously we have a very strong history and deep care about uveal melanoma in particular, given Mark's experiences. And one of the major ways that we've tried to action that mission is, as was mentioned, through a platform that we call Pattern.org. And so, I think this is not news to any of you, but I have it in here as a little bit of background. Even though we talk about rare cancers, certainly uveal melanoma among them, as individually rare, if you add up all the different rare cancers, it makes up estimated 1/3 of all new cancer diagnoses, right? So yes, individually these cancers are rare, but together this is a huge unmet need in the oncology space. And unfortunately, as I think was even hinted at in the last talk about FDA and approval and drug development, we have limited information about these, right? Because they're individually rare, a lot of times data hasn't been collected about how they progress. We haven't done the molecular analysis to understand what causes these tumors or what their vulnerabilities are. Whereas if you're in a more common tumor type, there are large research programs and the numbers are high and the data is available. So the goals of the Rare Cancer Research Foundation and Pattern, our program, and the kind of work we're doing with the Cure Insight is to try and break down that barrier to new progress by creating collections of data and samples for researchers to actually explore and understand the biology of these individually rare but together largely understood cancers. So I think I already sort of said this, but we don't have limited numbers of patients at individual institutions make it hard to, for instance, enroll trials or even generate hypotheses. The places that tissue is being collected and stored at some of these hospitals and some other research institutions often doesn't get utilized because it doesn't get connected to researchers who actually want to use that tissue and those samples to do experiments, right? It might sit in the freezer or the closet for a long time, and then, unfortunately, these things often get forgotten about. Additionally, researchers, right, may have interesting ideas of the kind of science they want to do, but they may not be in a place where they have access to those patients, that tumor tissue, or that data. you're a uveal melanoma researcher, but you don't work at one of the major hospitals that sees lots of uveal melanoma patients, you're sort of stymied. And so it's a little bit of a chicken and egg problem, because we want to encourage more researchers to work on these hard problems in underserved communities like uveal melanoma, but they're gonna say, well, that seems interesting scientifically, but I don't know how to do that because there aren't uveal melanoma patients at my hospital, right? Sort of similarly, and I know we've talked at length about this, one of the biggest tools that researchers use in preclinical development of drugs and targets is we call model systems. So these might be mouse models, they might be cell lines, organoids, but basically they're tools in the lab that replicate some amount of behavior of the tumor in the body, where we can poke and prod them, right? We can try drugs on them, we can run high-tech experiments on them to figure out what's happening in the biology of those. Those models are really the cornerstone of the beginning of trying to figure out what could be a new drug or what could be a new target or a new vulnerability of a tumor. They tend to be very sparse on the ground in rare tumor types, right? I mean, I can go to lots of resources out there and say, hey, I need a non-small cell lung cancer model, and there's 25 different options for me, and I can get them tomorrow shipped from 5 different labs in the country. That's not the case when you're talking about rare cancers, right? Because all of this other problem that we already talked about. And then I think, relatedly, a lot of people, as we sort of move into the age of personalized medicine and really understanding the value of data analysis, bioinformatics, personalized genomic testing, the same problem happens with data, right? We need large databases of sequencing data and clinical data and associated outcomes so that we can build models, in this case that means virtual models, right, of how that data can be interpreted to help patients and come up with new drugs and new options. So for all those reasons, we created Pattern.org. Pattern.org is a platform that allows patients anywhere in the US to donate living tissue and or data that we will de-identify and put out into the research ecosystem as much as possible. So the way we do this is, this is why we're doing this, right? I can come back to this. But the way this works is a patient finds out about us, they can register on Pattern.org. It's a totally virtual site. You don't have to come in, nothing like that. You consent to our research protocol online, sign a medical record release, and then we coordinate collection of tissue and or data from that patient. So if you're having a surgery, we can actually contact the hospital and say, hey, if there's extra tissue after the surgery, right? So number one, this doesn't affect clinical care, this doesn't affect the decisions that the doctor's gonna make. This is strictly where a tumor might be being removed and there's more tumor tissue there than they need for the actual, pathology, downstream clinical use. We can request they send that excess tissue to us, where we have the ability to capture it, we can freeze it, we can preserve it in a way that it stays living in the freezer so that we can bring it back out and researchers can try to grow it, and that's exactly what we're doing with the Cure Insight biobank. But in addition, we know a lot of patients either aren't having surgery because their particular case is not surgically viable, or they've had a surgery years ago, right? But they didn't hear about us. So this is not limited just to tissue. We also allow patients to donate their medical records. And the process is very similar. You sign up on Pattern.org, you consent, and then we sort of handle it from there. You have to sign a couple of forms to allow us to request your records, but then we go out, we contact the hospital, we get those records, and then within our HIPAA approved and secure systems, we remove all the identifiable information, we pull out the stuff that the researchers want to know, like what drugs did this patient have, in what order, and what was the result of each of those drugs, and maybe if they've had sequencing tests, what was the mutations that were identified in that sequencing, what was the gene expression pattern. And then we put all of that together, and we are building what we're now calling the Pattern Data Commons, the idea being that we can gather all of this de-identified data, put it in an open source repository where researchers anywhere can request access to mine that data, to look for new vulnerabilities in the tumor, to look for new possible targets, to try and identify similarities between uveal melanoma and some other tumor, right? I mean, it's not an accident that we call our program Pattern. one of the things we really strongly hope is that we're able to find linkages between these rare cancer types that aren't previously known, because of course, who's to say that a drug that's already been approved in lung cancer might not work for some subset of uveal melanoma patients? We won't know until we look and try to find those patterns. So those are the kinds of questions that we're hoping to sort of bring all of this data together into a place where researchers can start asking those questions. So ultimately, I know I talked a little bit about tissue donation, and certainly with the biobank and our historic use of pattern, that remains a huge focus for us. But I also want to highlight that in a lot of ways, the reason we need tissue is so that we can generate more data, right? The tissue by itself doesn't solve the problem, right? We have to get that tissue into the hands of researchers who are going to run experiments on it and generate information about how those tissue samples behave, how those models work, what drugs they respond to or don't. And then we want to capture all that data right back into this same collection of unified information that comes from patients donating their data, that comes from researchers participating with us and bringing us their sequencing data, and back and forth, right? We see this as sort of a bi-directional set of interlocking capabilities where these all feed on each other. So as I said, I can talk for hours about this, but I also know that there are two other speakers, and I also want to leave lots of time for Q&A. So I have some handouts and cards. You can find us on pattern.org. You can also the Rare Cancer Research Foundation. You can email me. I'm marshall@rcrf.org or info@pattern.org. Please feel free to participate. Please feel free to let folks know about us. I mean, this is a situation where our biggest challenge is recruitment, right? This works when lots and lots of patients throw in together, and the doctors too, right? Oncologists start talking to their patients about why this matters, and foundations talk to their patients, their communities about why this matters, and we get larger groups of people participating, and that's what eventually gives us the volume of data and capability that we need to find the next answers and the next problem to these serious problems. So that's sort of what I have to say, and I'm really looking forward to the Q&A, and I'll be here too. I can stay after the talk if anybody wants to chat.
Thank you. Q&A with Ryan. People have always, well, they've asked me, why do we partner with Pattern? Rare Cancer Research Foundation has been one of our groups that we've worked with over the years, and we've been watching this, and we've been helping to promote this with them for years. And when we decided it was a biobank, we looked at all kinds of platforms. And we just thought, why are we going to reinvent something and take all that time, money, and effort when they have a great system? So that's why we are partnering with pattern.org to do the collection and house the data. Our cell lines will also be grown at another lab, but we're going to be coordinating this with Pattern. But then there's another part of this that matters, is because patient data differs from physician input data. So what your interpretation of your medical record and what your physician puts in your medical record do not always match. So this has been a kind of a puzzling question that we've had. How do we figure out what patients understand their physician is saying, and how do we get that correct information that the physician is saying into our registry? The Data Commons is going to do that, but also we have another organization that we work with, Pulse InfoFrame, and they are doing the Omni registry. This is a registry that has been going on since, I believe, Ryan will clarify, 2018 I think is what they said, and they work with the oncologists. And it's a registry of physician input data directly from oncologists. Now, a Cure Insights registry is going to be able to talk to Pulse InfoFrame registry. So now we just added that extra layer. Now we've got your interpretation of your disease. We possibly could have some kind of biobanking material. We can get your actual medical record, and we can get now what the physician has put in that says this is what their interpretation of your disease is and what they're putting in your registry. So now we've hit it from all sides, and nobody else is doing this. So this is really exciting for us. So I want to introduce Ryan with Pulse InfoFrame. We've been working with them for a couple of years, trying to get our registries together. We share data dictionaries, we share information. All of this is in the consent that people have signed, but it's all controlled. So even though you're Mary Smith in our registry, you are probably patient 24839, and that's all that you know. That's all that anybody's going to see is what your patient identification number is. So we're going to hear from Ryan now. He's going to talk about Pulse InfoFrame and what they're doing over there.
Thank you, Melody, and hi everyone. My name is Ryan Sheedy, and I'm with Pulse InfoFrame. I'm the Associate Director of Business Development and Marketing, and I'm just going to take you through a little bit about what we do with our registry and how it differs from the one that Karen Insight is running. So I'm just going to share some slides here. But whoever's back there, just enable the sharing feature there. Thank you. Okay, so thank you again for having me in. As I mentioned, I work on the business development and marketing side here at Pulse InfoFrame. I've been with them for about 4 years now since I graduated college, and have been collaborating with Karen Insight and their team in varying capacities on different projects for about 2 of those years now. Now, I'm honored to join this conversation today about how data collection and now biological samples through the biobank are going to be helping us all move ocular melanoma research forward. And today we're going to talk about why both registries and biobanks matter, and how data samples and real-world data that we're generating through our registries can connect, and how they can collaborate to ultimately turn this trusted information into faster, more meaningful progress for patients. So before I dive into our specific work in UV, I'll just give you a little bit of background about what we do here at Pulse InfoFrame. So Pulse is a healthcare technology company that's been supporting research in oncology and rare diseases and other complex conditions for over a decade now. And we partner with biopharmaceutical companies, research institutions, patient advocacy groups to make it easier to collect, share, and ultimately use this high-quality real-world data safely and responsibly. And this comes in the form of what we call observational research, so patient registries, natural history studies, and also external comparator arms that are used to complement clinical trials. And we do this using a cloud-based platform that brings together different types of information and data all into one place. And it's really comprised of patient-reported data as an area of focus in some of our other registries with PROs, demographic information, and medical history. And then, as Melody was saying, clinician-reported data as well, so biomarkers, tests, procedures, and we are also able to integrate EHR data, imaging, wearables, claims data, and other medical devices as well. So I want to talk just briefly about just why data matters from such a high level here. So essentially, it's important to remember that behind every data point is a unique patient story, and every detail that you share, from when you were diagnosed to treatments you received and how they make you feel along the way, adds an immense amount of depth and understanding to people trying to study uveal melanoma. And each piece helps to complete the puzzle of how this disease behaves differently and how it affects people differently as well. And when we're able to combine these stories, we're able to see patterns appear easier. So researchers can identify what treatments are working the best, where complications are arising, and where there's still unmet need that we need to improve. So your individual experience, when it's viewed alongside others, becomes powerful evidence that shapes the future of care. Then ultimately, this entire process transforms your lived experience into actionable knowledge, and helps make more informed decisions, whether it's on the treatment front, better care patterns and approaches. And in that sense, every data point truly does matter, and every contribution is helping move research forward. Now, as Melody briefly touched on, our registry is different from Care Insights. So one common thing that I've run into a lot in my time here at Pulse is when someone sees that there's two different patient registries in the same condition, there's the assumption that we're asking the same questions and we're essentially duplicating efforts, when in reality it's not always the case, especially in this specific instance of UVO. So while there's some overlap areas, the Cure Insight really does tend to focus more on the patient experience. You can join online, diving into areas like quality of life, emotional well-being over a period of time. And again, this is incredibly important because it gives us a true perspective on what uveal melanoma really looks like from the patient perspective, which isn't always captured in a clinical trial setting. Given that there's strict eligibility criteria, it gives a much better real-world understanding, is what we refer to it as, of the patient experience. And our registry, we call it OMNI, which stands for Ocular Melanoma Natural History Study. We're collecting data from clinical sites all around the world, and it's continuously expanding each and every year in terms of not just the amount of patients in there, but also the geography we're covering in terms of our site network. So we're collecting it directly from doctors and care teams treating patients, and this includes details on tumor characteristics, treatment patterns and outcomes, test results, and as well as some patient-reported data that we collect through validated surveys. So overall, while both these registries are in the same condition, we're not duplicating efforts. They truly complement one another, which is part of what makes Cure Insights a great partner. And together, we're trying to provide a more complete understanding of the disease, how patients both experience it in their personal lives, and how it evolves in a clinical setting as well. And when we're able to combine these two perspectives, it truly helps drive progress, helps researchers develop better treatments and improve care strategies, and make sure that every study going forward can accurately reflect both the science and the patient voice in their design. So again, talking about collaboration is a great thing. I'm sure it's always a common theme at patient meetings and anywhere that you're really listening to, but how is it actually done in practice? So one of the most powerful features in our registries is called the Global Unique Identification Number. It's commonly referred to as GUID. So it's a secure code that keeps your information linked safely when it comes from a different source, like a patient registry, a hospital, or a biobank. So a GUID is created locally, and it never includes your name or personal information. It simply ensures that if you're participating in multiple studies, your data can be connected in a privacy-preserving way. And this makes collaboration possible without compromising anybody's safety or anybody's identity. Now, this is a long slide, but I'm just going to touch on it very briefly. I'll share kind of a real-world example of how we started using the GUID in another rare disease a number of years ago before I even joined Pulse. So, in this example here, this disease, there's lots of different data collection efforts ongoing at the time when we started, but none of it was able to connect, and it led to a lot of duplicated records, misconnections, and ultimately a lot of siloed data that was being underutilized. And for context, this disease is called CDKL5 deficiency disorder. It's a very rare form of epilepsy that affects about 1 in every 60,000 children, and it causes severe neurodevelopmental delays. Now, by implementing a GUID system, we were able to create one consistent anonymous code for each patient that allowed us to link multiple data sets safely. And this created an environment where we were able to bring in 7 different sponsors to all access the data at once in the same registry, Now, the overall result here was clear, more complete, and data that was ready to be used for regulatory bodies as well, all while maintaining privacy, and this is now the same approach that we're using for any collaboration that we do going forward. So whenever we're talking about sharing data, privacy is naturally going to be the first thing that comes to mind, and it really should be. It's a priority. It really should never be an afterthought. And before any information is shared, all personal details are removed, as we've talked about already. But this means your name, contact information, where you live, anything directly identifying you as a person is going to be stripped away. And what remains is the identified data set, and this information is all still incredibly valuable for research. It's just not traceable back to you personally. And both Insight and Omni follow some of the strictest privacy and data protection standards in the world. Again, HIPAA in the US, GDPR is very important for us as well, since many of our sites are based over in Europe. And they also undergo strict ethic board reviews to ensure that your rights as a participant are always protected. And again, it's important to remember that participation is always going to be entirely voluntarily. You're in control. You can choose when you join, you can choose when you withdraw at any time. And just as importantly, your data is never sold to third parties or used for purposes outside of approved research. And again, registries like this exist to advance science, not just for the sake of advancing it, but safely and responsibly. And when data is shared for research, it's done through secure and regulated channels. Now again, the big outcome here is that this allows researchers to see the bigger picture, connecting patient-reported experiences and clinical outcomes, all while maintaining complete privacy. So this allows you as a patient to feel confident that your participation is not only meaningful, but more importantly, safe. You're contributing to progress while keeping full control over all your own information. Now again, just touch here to kind of summarize some things, but having multiple registries is not duplication. In this case, it really is a strength. So given that each registry is collecting unique types of data, and these different data sources are able to collaborate in a secure environment, we get richer and more reliable evidence that benefits everybody. So we're able to look at the same disease through multiple lenses that adds a great amount of perspective, clarity, and depth. So again, even though these registries are focused on the same condition, they truly complement one another. And this gives researchers, clinicians, and advocacy groups a more complete understanding of uveal melanoma. Now, just to touch on the impact that you're truly making when you decide to participate in either a registry or a biobank, you're doing much more than sharing information on a surface level. You're helping researchers see patterns and connections that simply otherwise wouldn't be visible, especially in a clinical trial setting, again, getting back to that real-world understanding that we're looking for. And every data point is, every sample helps fill a different piece of the puzzle that we're trying to solve here. It leads to faster discoveries, stronger evidence, and ultimately better treatments. And for rare diseases like uveal melanoma, where every patient experience carries immense scientific value, these contributions are especially powerful. And your participation also strengthens advocacy. It gives groups like Cure Insight and organizations like Pulse the evidence that we need to push for better research funding, faster access to new therapies, and more personalized care options. So in other words, by choosing to take part, you're helping to change the trajectory of the disease, not just for yourself, but everyone else who's going to face it in the future. And again, just a few notes on how to get involved here. You can share your experience through Cure Insight's registry. Ask your clinic if they participate in a registry as well. It might even be ours at Pulse InfoFrame. And also continue to learn more about Cure Insight's new biobank program. And it's important to remember that every participant is adding value here, and every data point helps move the field forward. And looking forward, this really is all about securely linking patient voices and clinical outcomes and biological insights so we can have a better picture of the disease. And when all these data sources can talk to one another, we help accelerate research, and we're going to benefit more people in the long run. And that's the direction that we're all heading together. So I'll just close out with a couple of points here. So every data point matters significantly, especially given in a rare disease setting. Each registry entry, each donated sample, each shared experience adds another piece to the puzzle. And the more we share, the more we learn. When researchers, clinicians, and advocacy groups work together, we uncover insights that lead to earlier detection, better treatments, and improved quality of life for everybody affected by this disease. And before we open it up to Q&A, I want to just give a shout out to Cure Insight for inviting me to be with you all here today, and just for the work that they're doing every day. I mean, in my time here at Pulse, I've worked with several different patient advocacy groups and across a variety of different conditions, and we truly say that Cure Insight stands out as a model for success in what exceptional patient advocacy looks like. Not only their commitment to empowering patients and advancing research, but making a concerted effort to actually build partnerships across the community is just incredible work. And I'm always honored to collaborate with such a great organization. So thank you all.
Thank you, Ryan. Okay, so we kind of gave you an overview of what registries are and how they work, and that, data is important. So let's do a little bit of a deep dive into the data. Okay, so I'm gonna throw this question out to Ryan. Ryan, when you are in the Omni registry, what is the data that you're seeing? When who is requesting this data from you, and what does a typical request look like?
No, that's a good question. So typically, the data requests are coming from a network of, oh, sorry, I just see myself there. We work with a handful of different sponsors that have access to the registry as well. So specifically, they come in with a new research question that they're trying to look at is usually what the specific request comes in. They have access to what we call a dashboard, basically, that shows an aggregate view of everything that's inside the registry. And this is in a Tableau environment, so they're able to filter through different mutations if they want to address a specific subgroup within the disease and get the same kind of information that way. But generally, it's coming from sponsors that we work with, as well as, again, researchers have access to their own view of the dashboard as well too. It's a little bit different for them if they're coming from a site, but those are really typically the two requests that we're working with when it comes to data. Usually for the purpose of their ongoing research, but also for publications as well. There's been a lot that's been generated from Omni.
Great. Now, Marshall, what are you seeing? What are researchers asking you for? What are you seeing that's the hot thing? Oh, can we turn the microphone?
Oh, there we go. Actually, very similar to what Ryan said. A lot of my background, I trained as a geneticist, genomics. So one of the things that we're trying to highlight in the Pattern Data Commons is really linking high dimensional, rich molecular data with the clinical annotation. So part of getting the medical records, as we do, is we can also find out whether a patient has had a sequencing test, right? So many of you are probably familiar, there are Tempus and Boston Gene and Caris, there are lots of these vendors out there. Because HIPAA, the regulations say that any clinical test results that are done on a patient belong to that patient, we're also able to request from those vendors the raw sequencing data that's generated by those tests, right? So when you get your Tempus test back or your Boston Gene test back, it's a PDF, it goes to your oncologist, it's structured in such a way that's organized and oriented toward clinical care as it should be, right? It gives you potential likely drugs that might work, or information about prognoses based on your genomics. But the actual test that they run in the lab tests lots and lots of other things, right? A lot of those tests are sequencing essentially the whole genome of your tumor. A lot of that information we don't really know what to do with clinically yet, but that's the information where eventually we're going to find new targets, we're going to find new vulnerabilities. But that's a research question, right? So certainly your oncologist doesn't need to see that data. That wouldn't be particularly useful for them. But one of the things we're doing, in addition to the sort of what's in the medical record, is trying to capture as much of that deep molecular profiling data and linking them together so that when researchers ask these same kinds of questions that Ryan's mentioning, we can layer onto that, hey, there's a bunch of sequencing data about this tumor as well. Maybe that's something you want to correlate with the kinds of questions that you're asking of any of these registries.
So when we start these three biobank registry platforms, when we can get these linked together, and a researcher says, I would like to have three cell line profiles, and I would like to study somebody 30 years old and under, and I want to study any females, and I want them to be a class 2. So we would take our registry data, we would look to see if there's any biobank data, we'd connect with Pulse and see if they have any data. Now they have this profile. Now what? Now what happens? Where does it go, and how do they use that?
That's a really good question. And I think the answer is it depends a lot on who they are and what they're doing, right? So a lot of and I'd be interested in Ryan's perspective here too, because I think Pulse sounds like he's working a lot more closely with sponsors lot of the folks who I work with on more of a day-to-day basis are academic researchers, and I think that there's an important collaboration there, right? I mean, most drug companies aren't doing what I would call very early stage target discovery preclinical work, right? That's sort of been the purview of academic research for the past 25 or 30 years, and I think that will probably continue to be the case. So in the case that you described, my imagination immediately goes to, well, that's somebody with a lab at a research university or an independent research institution like Fred Hutch, where they have some smart bioinformaticians who might be postdocs or graduate students, and they're going to take all that data, and it could be terabytes of data if we have sequencing, for instance, or if we have cell lines, we could do the really crazy high-dimensional immunofluorescence and like really exciting new lab techniques. They're going to take all that data and they're going to comb through it with sophisticated statistical techniques, or maybe they're going to apply new deep learning models against it to try and identify correlations with outcome, right? So like that's why we need the registry data. We say like, well, we don't know why this patient had a better response than that patient. How can we find those correlates, and what are the drivers down in the molecular data that hint at why that might have been or might not have been? That's sort of my vision for the kinds of experiments, the kind of use of that data would be. But I actually would be really interested in what Ryan thinks on sort of the more, as you go further into the sponsor commercialization, drug development side.
No, yeah, it's a great question. I would say, again, it really comes down to who's asking and also where are they at in terms of clinical development of their asset. So one common use case is using the registry to build external control arms. So for people who don't know, this is something that you use basically complement clinical trial data, because since UVL is a rare condition, it's not always seen as ethical to use a placebo, essentially. So what you can do is essentially build an external group that's outside of the clinical trial, but that largely mirrors their enrollment criteria through the patients that we have in our registry, and compare what they're seeing in their clinical trial alongside just people completely removed from the clinical trial as well. So that, I would say, is probably like one of the biggest use cases for Omni, especially just in terms of complementing sponsor activity. But another one too is just helping complement them with natural history data. So there's a growing trend now too. We actually just put out an article about this recently of sponsors more proactively trying to run natural history studies in advance of going into clinic, so that they can ensure that they're hitting correct endpoints, so they can make sure they know as much about the disease as possible. And it essentially helps kind of de-risk their entire endeavor trying to develop this drug. But no, that's a great question.
Okay, so we're looking at now, we've got data from two sides. We've got the academic. These are the people that are in universities and that are doing research. But then we've also got the sponsor side, the pharma side. So they don't necessarily want the same data for the same reason, but it's going to come together to make an end point. How would this data benefit a pharmaceutical company sponsor more so than an academic researcher? Because we have heard, Karen Site has heard, oh, you don't want to have these cell lines go to pharma. No, they need to stay in the academia world. No, that's bad, don't do that. And we're like, but wait a minute. How is pharma going to advance UVL research if they don't have any cell lines themselves? Because that's the whole point. We don't have enough cell lines. So how do we, help us with that argument?
I mean, the slightly pithy, but I think still accurate response that I have to that is, if you want new drugs, pharma's going to have to be involved, right? I mean, for better or for worse, and I think as we saw in the previous section, right, the pharma folks want to help patients. That there's a lot of, political and sort of, I don't know what the right word is, but there's a lot of angst about how money works in that system, and it's a very complicated system, and I'm not going to pass judgment in any direction on that. But the fact of the matter is, they're the ones who will make the drugs, right? So ultimately, if they have a good question that they think a cell line might help them get closer to getting a drug onto the market, I'm all for getting them cell lines. Certainly the question of IP and when you're in the academic space, like who gets authorship on the paper, right? Or how does the university that paid for the salaries of the workers, maybe it's grants, but like how does the university who sponsors the lab that this work was done in, they have a vested interest in the ultimate financial return on that drug, maybe. So, but this is not atypical, right? Most drugs, early stage targets, are being discovered in academic labs, and ultimately part of the money that is generated by the sale of those drugs eventually does end up back at those universities through IP relationships and licensing, right? There are armies of lawyers who take care of that stuff. It's totally not my specialty, but like this is not, that collaboration between academic sites and pharmas is not new and it's not surprising, and we should, I think, embrace it, especially in rare cancers, because we know we need more options for patients. And if you can get the pharmas excited about it, they're the ones who have the infrastructure and the experience and the capability to make those options real.
Do you have anything to add, Mike?
Not much. I think Marshall just summed it up perfectly, really. I think a lot of it is just, again, maybe a point in terms of, again, pharma does genuinely want to help patients, again, at least, again, especially in rare cancers, and especially just given the nature of it being rare, you kind of just need to push that collaboration. It's really just more about looking forward. Again, I think Marshall summed it up like perfectly.
So if somebody's going to donate information of whatever kind it is, whether it's medical records, registry participation, or biosamples, what is the best, what is the best outcome for that patient to be able to say, hey, I donated this, now I've done that part, now they've taken it over, and you need to communicate back to the patient the value of this donation that they've made. While we can't go back and say to patient A, hey, your data was used in this study, we can't report back to you about that, but what message can we give patients now to make them feel really confident and really comfortable to give their data, and then to also to get them to understand the contribution that they're making to science and to advance uveal melanoma research going forward?
So maybe I'll take those questions a little bit in reverse and answer the last one because I feel slightly more equipped to do so. The science can only happen, right? Like we're very limited as scientists, especially, my background was in studying gene expression, so the way cells turn on and off different genes. And we used I used a lot of cell lines. These were cell lines from like the 50s, right? That were generated before sort of our modern structure of like patients consenting the way they do. And it was useful, right? Like we found out lots of cool, interesting stuff, but it was very limited in terms of, okay, how generalizable is this? This cell line has been growing in the lab since the 1950s. It probably really doesn't look like the cells that it started as, right? They've mutated over many years. Furthermore, I actually don't know anything about the clinical history of the individual that these cells were derived from. So it was sort of a bag of genes that had some useful capability for like general research, fundamental basic biology. But I couldn't have said anything to you. They happen to be derived from T cells. There's no world in which that cell line could have helped me discover something meaningful about T cells in a particular cancer type, right? They were just too far removed from the actual clinical and patient. And that was not that long ago. I mean, it feels like a lifetime ago, but the story hasn't changed that much. So we need better annotation of those kinds of tools in the lab. And by annotation, I mean understanding this came from a 37-year-old individual who had been treated with radiation and chemo, but not, IO or whatever the specifics are, to really get down to understanding why those cells are behaving the way they are. And that only happens these days when patients are willing to donate this combination of samples, of data, of their sequencing information. So the bar, unfortunately, is so low right now in rare cancers that, yes, you're right, I can't turn around and tell a patient like, hey, that Nature paper that just came out, you were patient 375 in that paper, right? Like ethically, that's sort of outside the bounds of what we as researchers feel comfortable with, and the IRB agrees. But I can say that in these rare cancer types, patients who donate tissue and who donate data, that's going to be meaningfully impactful, within a couple of years. And if you follow the literature, you can be sure that those papers would not have come out or would not have had the ability to make the conclusions they've made without each one of those patients contributing to that study and that finding.
And Ryan, you have anything to add?
Not too much to add there, but I would say, again, it's, as much as we would love to be able to say, your contribution helped X company do A and B kind of thing, it's just not really something we can do feasibly. However, I think it's more important to just kind of step back and take, again, big picture, it's a rare cancer, and again, every little bit just helps so much more than we can really put into words, at least like in a, like a common way. But I would always just try to encourage patients to stay up to date on the literature, whether it's, and a good way to do that is even just following a current site closely. I know they're great about just sharing everything online, and that's a great way just to stay. Again, you can see these academic papers that might come up, and while it might be hard to wrap your head around some of what's actually in there, if you follow it through just an advocacy group or any kind of more patient-friendly channel, you can kind of see that, okay, it's going somewhere. But the big thing is that we always try to say is how can we show patients that when they're donating to a registry, it's not just going in a black box and just kind of being stored away. And again, we've tried to at least show that through, okay, we're generating publications. But again, it's more just kind of taking a step back and seeing, okay, there's progress being made, and you're part of the picture when it comes to that progress, in short.
The big movement right now seems to be in patient-reported outcomes, patient involvement in clinical trials, patient input, like we had our roundtables with pharmaceutical companies to help develop drugs. The patient voice is huge, and while organizations and research loves donations of money, we would love donations of your information. It's probably more valuable than a donation that you could make in cash for a research project. A lot of research projects can get grants and then get funding, but they can't get your information. We can't produce cell lines out of thin air. We have to have viable tissue in order to make those. We have to have medical records, both patient input and also physician input, to put this story together. With the story, putting it all in a box and coming out with a profile. I use the box as a term loosely because I'm not computer literate, but Marshall's probably thinking, we don't use a box. But it goes in these wonderful computers and it comes out and it spits out this great information. And through this and through the I don't know, better development of software and computer technology, they're going to start finding more and more stuff, because 5 years ago we didn't have a lot of the stuff we have now. We just didn't have the ability, and now we do. So this is a huge thing. It's a huge way to get involved and to give back and to try to get science to go forward. With, like Marshall said, we need updated cell lines. We really need tissue. We need updated medical records. We need all this information. So if your physician asks you to participate in a clinical trial on his side, please do. Please join the Insight Registry. Please consider donating something to the biobank. If you don't have tissue or blood samples or that sort of thing to donate today, you can donate your medical records today. Now that would help, because with the Insight Registry and with Pulse and the Omni Registry, and then with the Pattern.org bringing together the data commons, we could create something so powerful that has not ever been done before. And our little tiny community could really be trendsetters and really push this forward if we can get everybody to help us out and just donate something. It costs you nothing, absolutely. Like Marshall said, you can withdraw at any time. But if you don't register and put forth a little bit of pain to fill out the surveys or sign the consents, we're not going to get there. So in order for what Omni is doing, what Marshall and Pattern.org is doing, in order for that to work, we have to have patient input. We have to have you guys get on board with us and realize the magnitude of what can be, because it's going to be huge. You guys have any last words?
No, that was perfect. I couldn't agree more.
Well, we'd like to thank Ryan and Marshall for being with us today. Thank you so much. We're going to have lunch. Megan, you got some announcements? Or are you good?
Check out our store after lunch and check out the merch.
Yeah, we got merch store out. We got a photo booth is up. Take some photos. We did get the backdrop up from its resurrection of the crash last night, so we're good to go. So
some magic.
yeah, it's up. So anyway, go have some fun. Go ahead,
Can
Lauren.
I ask you a question though? If somebody's, you're looking for metastatic disease for the biobank, or is it
Anything,
anything? So
anything.
somebody who's, their cancer is,
Initial tumor tissue of
okay,
your eye.
yeah,
They're also going to take vitreous fluid
okay,
in your eye. They're taking blood samples. Anybody can donate blood samples today. And then if it goes metastatic and you have a biopsy of your metastatic tumor, they can also use that. But it can be your original eye tumor. We
So
cannot
what if
take,
somebody's, like you and I are way out there, our tumors are
but,
dead,
so we can donate blood,
okay,
okay? And we can donate our medical records.
okay,
Okay, they can get a lot through our medical, even though when Lauren and I were diagnosed, we did not have genetic testing, they really can't tell much, but at least they can go by our sex, our age, kind of our journey. That will help. What other diseases you've had? Yeah. Anybody else have any questions? I totally skipped over Q&A, I apologize. I got a little passionate there, sorry about that. Hi, everyone. All right, we're good? Okay, you
Thank
guys,
you.
please, Marshall's gonna be here. If you need anything, you can